Hello students, in this video I will be answering multiple questions. To begin with, the first question wants us to explain and mention various types of characters that are used by the clinicians to identify microbes. Clinicians use a combination of phenotypic, genotypic, and biochemical characteristics to identify microbes from the clinical samples. Phenotypic characteristics refers to observable traits such as colony morphology, pigmentation, and growth rate. Genotypic characteristics refers to genetic makeup of the microbes such as DNA sequencing or hybridization. Biochemical characteristics refers to metabolic processes and enzymes that the microbe use to break down nutrients. Moving on to the next question, three biochemical tests that are used to identify a microbe are catalase test, oxidase test, and indole test. Catalase test is used to identify a bacteria that produces the enzyme catalase. The catalase enzyme breaks down hydrogen peroxide into water and oxygen. Bacteria that produce catalase will bubble when hydrogen peroxide is added to their colony. Next one is oxidase test. It is a test used to identify the bacteria that produce the enzyme oxidase. The oxidase enzyme is involved in the electron transport chain and is used to identify certain groups of bacteria such as Pseudomonas species. A drop of oxidase reagent is added to the colony and a color change indicates the presence of the enzyme oxidase. The last one, indole test. This test is used to identify the bacteria that produce tryptophanase. Tryptophanase breaks down the tryptophan into indole, pyruvate, and ammonia. A positive result for the indole test is indicated by the appearance of a red ring after adding coax reagent to the bacterial culture. Moving on to the next question, environmental microbes use a variety of nutrient sources such as organic matter, inorganic compounds, sunlight. Organic matter is composed of plant and animal material which provides a source of carbon and nitrogen. This includes dead and decaying plant roots, leaves, and animal remains. Inorganic compounds. Some environmental microbes use inorganic compounds such as ammonia, nitrate, and sulfate as their nutrient sources. Sunlight. Photosynthetic microbes such as cyanobacteria use sunlight as a source of energy to produce organic matter through photosynthesis. Moving on to the next question, to encourage the growth of soil microbes and plant growth, various chemicals are added to the soil. These include fertilizers such as nitrogen, phosphorus, and potassium, as well as soil conditioners such as lime, gypsum, and organic matter. Moving on to the last question, antibiotics and disinfectants both kill or inhibit the growth of microorganisms, but they differ in their mode of action. Antibiotics target specific bacterial cells and disrupt their metabolic processes, while disinfectants are non-specific and target a broad range of microorganisms. Antibiotics work by interfering cellular processes such as bacterial cell wall synthesis, protein synthesis, DNA replication, while disinfectants work by damaging cellular components such as cell membrane, denaturing proteins, and oxidizing cellular components. Additionally, antibiotics are used to treat infections in living organisms, while disinfectants are used to sanitize surfaces such as equipments in hospitals, laboratories, and in other settings. I hope you find the video useful. Thank you.